Hola, muy buenas tardes a todos. Estamos en directo. Hoy se celebra el Día Mundial de la Inmunología, en realidad es mañana. Y como esto se han programado diferentes eventos alrededor del mundo. En Colombia, en nombre de la Asociación Colombiana de Inmunología, le damos la bienvenida a todos y hoy tenemos una conferencia realmente excitante. Solo quiero hacer la introducción inicial, después el doctor Federico Perdomo hará la introducción de nuestro conferencista, que ha sido un referente mundial en el tema de la respuesta de linfocitos T contra infecciones virales, especialmente VIH, el doctor Michael Betts de UPenn. Así que espero que estén conectados, que disfruten la presentación y que haya muchas preguntas. Y muchas gracias por conectarse ese día. Así que Federico, adelante.
Okay, gracias, John. So we're going to move to English. So it's for me a great pleasure to introduce Dr. Michael Betts. So Dr. Betts earned his PhD in 1998 from the University of North Carolina, working on human T-cell responses to HIV. Then he continued his work during his postdoctoral fellowship with Dr. Richard Coop, first at the University of Texas Southwestern Medical Center and then at the NIH Vaccine Research Center. And finally, in 2005, he started his lab at the University of Pennsylvania. And he's continuing studying human T-cell function in order to understand the role of these T-cells in controlling or eliminating viral pathogens and providing protection from infection. Of course, the primary interest in his lab is determining how human CD8 T-cells can respond to HIV and other viruses. And just to let you know, his lab have a very funny and interesting web page, so I invite you to visit that web page. So Dr. Betts, welcome to this presentation, to this talk. We're very happy to have you here, to share your knowledge with us. And just to start, I would like you to say a few words about why it is important or not important to study immunology. So we have in our audience a lot of people, perhaps not immunologists. So I just want you to know your perspective about that. Welcome.
Thank you very much for the invitation, Federico. It's really nice to see you again and to meet everyone else who I've been able to meet virtually. So thanks for that question. I haven't thought about it in that sense, but I've always been interested in immunology in a selfish way. I want to understand how I fight diseases, and I think it's just fascinating to think about how our bodies constantly are dealing with threats of different pathogens or even commensal organisms all the time, and our bodies have to combat or tolerate or ignore these pathogens and all of these different, there's many different mechanisms by which the immune system has developed to protect us. And so we've studied CD8 T cells for a long time, and trying to understand their role, because I think they're some of the most fascinating aspects of the immune system in terms of what they do.
Okay, thank you very much. So I think we can now start with your presentation.
Excellent. Okay, I will switch over to full screen mode. Federico, can you confirm this looks okay?
Yeah, it's okay.
Okay, excellent. Thank you. Okay, everybody. So today I will tell you about new study in our lab, and then combine that with some data from a recently published manuscript as well, to try to describe our studies about CD8 T cells and what they're doing in HIV infection, but at the end, come around to what we currently believe in the lab as the most logical, if you want to say rational design of a therapeutic eradication strategy for HIV. And I won't say that we've figured out how to do it or been successful, but I think based on studies that we have done in part with the study I will show you today, I believe I have a different roadmap to try to get in this direction. So let's talk about HIV. So of course, with HIV, once individuals are on therapy and fully suppressed, we know that for life they could take antiretroviral therapy. But at any point in which they would stop taking ART, the vast majority of individuals will have a viral rebound, as shown in the center of the slide. And they will essentially return to the state of disease in which they had prior to initiating ART. So their viral load will rebound about to the levels of the initial uncontrolled infection, and they will then continue to progress with their disease. And the reason why this happens is because of what's called the latent HIV reservoir, and this is a persistently infected population of CD4 T cells, monocytes, and perhaps other cell types as well in the central nervous system and so on, that harbor latent provirus. And after removing therapy, that latent provirus can reawaken upon T cell activation or cellular activation if it's not a T cell, and then the viremia returns. And we know that the vast proportion of this viral reservoir is actually not found in blood, but it's really found in tissues and organs. And that makes it imperative to understand how to apply therapeutic strategies to the tissues rather than just simple procedures where one would try to cure somebody by only targeting cells in the blood. And one of the key components of control, and one of the primary targets that people wish to invoke for cure-based strategies is cytotoxic T lymphocyte activity. And that's because we know from a variety of studies that these cells play a very critical role in ongoing control of disease progression. It's very important to know that, of course, that although they effectively control disease progression, they are unable to eliminate infection, at least in any case that has been documented. So although they control, they can't eliminate, but that doesn't mean necessarily that they couldn't do so in a therapeutic context if provided with the proper tools. And so we know that CD8 T cells in at least in the monkey model of SIV infection, which is in fact what I'll be talking about mostly today, we know that if you deplete CD8 T cells in SIV infection, that viremia will rebound, so providing a direct indication that they're playing a role in control. We know in HIV-infected individuals that people who don't progress in their disease have enhanced properties of their CD8 T cells, and this includes heightened proliferative capacity, indicating probably a more stem cell-like CD8 population playing at least a partial role in ongoing maintenance, but also increased ability for these cells to either have or differentiate into cytolytic effector molecules, cytolytic effector cells. So the classic CTL, and a number of different properties associated with this type of differentiation. But it's really important to understand, and it's an unfortunate thing, that that CTL, the cytotoxic T lymphocyte, or the CD8 cytotoxic T lymphocyte, is often assumed to be the primary function of all CD8 T cells. So it's important to understand that not all CD8 T cells are cytotoxic. And much like CD4 T cells, CD8 T cells are composed of multiple functional subsets and differentiation states, of course, including the cytotoxic effector cell, which for this talk I will largely consider as those cells using the perforin and granzyme mediated killing activity, which is probably the most potent method that the cells can use to eliminate infected cells. But there's also resident memory cells, which would, of course, stay in tissues and mediate their function in the tissue environment. And then a variety of different other subsets, memory subsets, and so on that have an ability to patrol the body in different sites. So central memory go to lymphoid tissues, effector memories can go to lymphoid tissues and non-lymphoid tissues, and so on. And they just essentially patrol the body in an autonomous fashion, responding to inflammatory cues and mediate their function throughout the body. And one thing that's become very clear from the studies that we and others have conducted is that this functional properties differ very dramatically between the vasculature and tissues. And so the graphic on the left sort of documents our thinking on this in the context of HIV. And so cytotoxic CD8 T cells, in other words, those expressing perforin and granzyme, we know from, in the context of HIV infection, that they appear, an SIV infection, that they appear very early in infection in these tissues, but that they are lost progressively and rapidly from those tissues. We've never really quite understood why they're lost. The study I'll talk about today partially addresses some of that. But it's very clear that they're lost, and it's not necessarily due to cell death. It's not necessarily due to trafficking. There's probably more complex regulation taking place. We also know that blood CTL, the classic cytotoxic cells, are excluded from lymph nodes and tissues in the chronic phase of HIV infection, and that's because these cells don't express the proper adhesion and homing receptors to traffic into tissues. They lack CD62L, they lack CCR7, and this prevents them from directly entering the tissues where of course, the infected cells are residing. Once we're in the tissues where the infected cells are located, we also know from a variety of different studies that whether in lymph nodes or in non-lymphoid tissues, the CD8 T cells in those locations are poorly cytotoxic, and it's most likely that these cells mediate their function in a non-cytotoxic manner. In other words, they're not using the perforin granzyme mediated killing pathway, which largely will induce a very pathologic response in tissue damage if it's in place. And so the classic way to think about this is cutaneous leishmaniasis, when you have the lesions that occur in the skin because you have truly cytotoxic T cells in skin where they don't belong, and they cause tissue pathology, and in fact are actually quite unable to eliminate the pathogen anyway, but it's just the wrong function in that place. And we've figured out that by looking at the cells, whether they're in the blood, in the tissues, or in the lymph, which is the conduit by which all the CD8s would leave lymphoid tissues and go back into the blood through the thoracic duct, we've understood that there are different transcriptional and epigenetic regulatory states of these cells, and that even within a clone of T cells ex vivo, that there's differentiation such that some daughter cells of the clone will be non-cytotoxic and able to traffic into tissues, and other daughters of the clone will be cytotoxic and trapped or relegated to vascular surveillance. And so this is the way we think about CD8 T cell function sort of across the body. And when you think about these different points, you have to consider then how CD8 T cells are controlling the HIV virus, which resides in tissues. And so what we think about then is first of all, trying to understand how and why cytolytic HIV-specific CD T cells are lost after acute infection, and is this due to trafficking or regulation? Are we able to get HIV-specific T cells from the blood to enter tissues and become cytotoxic if activated? Because that would be the desired outcome, is actually just to induce cytotoxicity and kill the infected cells, whether they're in lymph nodes or in non-lymphoid tissues. And then ultimately, of course, can HIV-specific CD T cells that mediate their function in tissues control plasma viremia? Because when you think about what is the source of plasma viremia, it's most likely infected cells in the tissues that release virus that gets into the vasculature. So, and then of course, how do we address these questions in vivo, which is where we would really want to understand them. And of course, for this, we would use the monkey model. And so the concept of this study that I'll talk to you about today is that the failure to effectively control or eliminate HIV may be due in part to the inability to induce or maintain CD8 T cell cytotoxic properties in tissues. And so our goals for the study are to define the differential roles of tissue versus vascular CD8 T cells in the control of SIV infection, determine whether CD8 T cells in lymphoid tissue become cytotoxic after encounter with infected cells in the lymph node, and determine whether we can modulate these cells through therapeutic immunomodulation to enable cytotoxicity and control of viremia. And so our strategy is actually to in vivo manipulate T cell trafficking to determine whether the CD8 T cells acquire cytotoxic properties in tissues of infected animals and control viral replication during an antiretroviral therapy interruption. So I'll explain how this works next. What we're going to use is a drug called FTY-720, and this is a drug that's used clinically for the treatment of multiple sclerosis in some individuals. This is an inhibitor of the sphingosine-1 phosphate trafficking pathway, and this is shown in the center of the slide. So what FTY-7, I should say, lymphocytes traffic around the body in response to the sphingosine-1 phosphate gradient. And sphingosine-1 phosphate is produced by endothelial cells in the lymphatics and the vasculature, but not in the tissues. So essentially what happens is you have these endothelial cells in these places producing sphingosine-1 phosphate, and it's that gradient then is higher in the blood and lymph than it is in the tissues. And so the cells in the tissues, if they express sphingosine-1 phosphate receptor, will respond to this gradient and move into the vasculature space. And then once they're in the vascular space, they downregulate sphingosine-1 phosphate receptor, which allows them to move back into tissues. FTY-720 inhibits this pathway. And downregulates sphingosine-1 phosphate receptor from the cell surface, so thereby traps the cells in their tissue location because they can no longer respond to the sphingosine-1 phosphate gradient. And so all lymphocytes that are when treated with FTY-720, all lymphocytes capable of trafficking to tissues get trapped in those tissues. And I'll talk a little bit more about this in the model slide on the next slide. We've already published in 2020, interestingly, that the fully differentiated CTL in the blood, those perforin-expressing CD8 T cells, in humans do not leave the blood under FTY-720 treatment. So we know that if we treat with this, we will trap basically it's not really trapping because the cells are already trapped, but we will purify for cytotoxic T cells in the vascular space. So the combined effect of this will essentially separate the infected CD4 cells, which have a trafficking phenotype, will separate them from the CTL in the blood, and all those infected cells will become trapped in the tissues, where they will only be accessed by the tissue resident or the tissue migrating CD8 T cells. And so that's really highlighted here. Just again, I'll go through it again to clarify, because it's very important to understand the model for the way the science ended up working out. So as I mentioned before, CTL, this classical fully differentiated CTL phenotype, remains in the blood. And this is both for CD4s and CD8s. The migratory CD8s, such as central memory and so on, they will go into lymphoid tissues and to other tissues and come back to the blood. The infected CD4s have these same phenotypes and they migrate. Uninfected CD4s migrate. But then in the tissues, you have resident CD4s and CD8s. When we treat with FTY, we change this dynamic. And so the CTL will still remain in blood, but now all the other cells that migrate are trapped in the tissues, and this includes the infected CD4s. And then now will allow us to test whether the tissue CD8s are able to impact the infected CD4s, which are now locally contained within the same tissues. So the study design was, this was done in rhesus macaques. These are all MAMU A01. This is an MHC class 1 haplotype. They're all MAMU A01 positive animals, which allows us to use MHC class 1 tetramer reagents in the system to interrogate directly the SIV specific CD8 T cells. We set up the study with an acute infection in the animals, as shown on the bottom. This was using a SIV MAC239, a virus that we had barcoded so that we could track individual viral variants over time. I'm not going to talk about that aspect of the study here. We're just going to talk about what happened on the immunology side rather than the virology side. We initiated ART at day 14 post-infection, so very, very early. And this was done to preserve some of the T cell functional properties. The animals were all fully suppressed, and we kept them suppressed out to 220 days post-infection. In half the animals, we initiated FTY-720 at day 190, or 30 days before we were going to interrupt therapy. And so it's at the tail end of the graph on the bottom. Once FTY was started, you can see that there were no viral blips that occurred when we redistributed the lymphocytes using FTY-720. The FTY-720 was very effective in its properties, and so the graphs on the bottom right show the effect of FTY-720 in the animals. So just to orient you to the way, and I'll have a lot of graphs that look similar to this coming up. On these graphs, the line in red will always be animals treated with FTY-720, and the line in black will be the controls. The gray boxes are the periods of time when there was ART present, and the white area is when ART was interrupted. And you can see the period during which FTY-720 was kept in place. So we kept FTY in place for 4 months or three months, 120 days, 3 to 4 months. And so as predicted, the FTY treated animals, we see on the first plot on the bottom right, the CD4 T cells, we see almost a complete depletion of CD4s from the peripheral blood, whereas the control animals maintained CD4s in the blood, as one would expect. We also see something similar happen with CD8 T cells, and we'll talk more about that on the next slide. So the CD8 T cells also have a very marked depletion in the peripheral blood during the FTY-720 treatment. But it's not all CD8 T cells, as you can see on the graph on the left. Although they decrease on FTY, they don't become zero, and that's because, as I mentioned earlier, the effector and effector memory subsets, and these are the CD8 T cells that express perforin and granzyme, as shown on the right part of the slide. These cells do not traffic out of the blood, and so basically the cytotoxic cells become purified in the blood, and everything else is gone from the blood. So they're basically in the blood with no targets, and nothing to do. And you can see that you really have a very purified perforin and granzyme population present in the blood in every animal. If we look at the SIV specific cells, we see the same thing happen. So although a lot of the cells, of course, traffic into the tissues, here showing on the top left, the TL8 and CM9 specific CD8 T cells, these are the SIV specific T cells in the blood of these animals. If we look on the top right, before FTY treatment, only a small proportion of them are expressing perforin and granzyme B, but after FTY or during FTY treatment, you can see a massive shift of these cells so that the vast majority of them, and that particular frequency shown in that example flow plot doesn't quite do justice to the obvious that everything is becoming quite positive. And you can see in the bottom, again, that during the therapy interruption under FTY, we get a very strong SIV-specific cytolytic effector purification happening in the blood. We then looked in lymph nodes, because this is where we really wanted to understand what was happening. And the first thing is, and this was done by immunohistochemistry, working with Claire Deliage at NCI Frederick. We first looked at localization of CD8 T cells in the lymph nodes, because now the trafficking CD8 T cells have gone into the lymph node, they're trapped there, and the question is, where are they going in the lymph node? Are they going to go to the places where we know that the virus is present, which would be, first of all, B cell follicles, where the infected T follicular helper cells are present? And then alternatively, are they in the T cell zone? And so in the red line here, whether it's in the B cell follicles or in the T cell zone, we can see marginal increases of CD8 T cells per tissue area. And in the T cell zone, after prolonged FTY treatment, we can start to see an increased level of CD8 T cells present in the lymph node. We can also see during ATI that some of these CD8 T cells that are trapped in the tissue are beginning to get activated and be cycling, particularly after the time point in which viral rebound occurred, shown in the center of the plot. We also looked in gut tissue, and we see something similar, a slight increase in CD8 T cells in the particular biopsy that we looked at. But if we look in follicular aggregates, so this would be the lymphoid tissue follicular aggregates that are present within the gut mucosa. But if we just look at the gut mucosal tissue itself, that we could not see really a change under FTY in terms of the number of cells per area. We also saw a very small increase, but actually not different than controls in cycling cells as measured by Ki-67. We then, because part of the study was designed to ask whether there's a difference in T cell residency during FTY, we looked at CD69 expression on the SIV specific cells, and we did not see that FTY-720 increased residency formation in the CD8 T cells lymph nodes. In fact, the control animals seem to have slightly higher levels of CD69 compared to the FTY-720. And we also did not see an enhancement of follicular homing SIV specific cells as would be judged by CXCR5 expression. So although FTY-720 is absolutely trapping cells in the lymph nodes, and we can see a slight numerical increase in them, we aren't seeing an increase in residency, and we're only seeing sort of small changes in the homing properties into the HIV infected follicles. We also confirmed this lack of a TRM phenotype by doing transcriptomic analysis on animals with or without FTY-720. And I'm not going to show that data because it would require too much time to go through it. Just to keep it simple, we did not see that the non-resident cells were, when trapped by FTY-720 in the tissues, were acquiring a signature that looked like a resident cell. So the drug doesn't make a non-resident turn into a resident. But really the important question was what happens to the cytotoxic properties? Because remember, at this time, we've interrupted therapy. The virus, as I'll show you in a moment, has rebounded in these animals. And so now you have a situation where you have the CD8 T cells trapped in the tissues, you have the infected cells trapped in the tissues, and you're letting the virus start to replicate again because they're no longer on therapy. And so what we had hoped for, the hypothesis was that this proximity and keeping the cells together with their targets was going to make these migrating or tissue resident CD8 T cells differentiate into cytotoxic T cells. And as you can see here, as measured by perforin, and this is looking actually at the total CD8 T cells, not just the SIV specific ones, that we do not see an increase in perforin expression during the therapy interruption on the FTY treated animals. And we also looked and found also no substantive change in perforin expression in the gut mucosa either, as shown on the right. So just exposing the CD8 T cells to antigen in the tissue is not changing their inherent properties of being non-cytotoxic into a cytotoxic cell. We further tested this by using a redirected killing assay. So here we take purified CD8 T cells from the tissues of interest. We in vitro incubate them with a target cell that's just simply coated with anti-CD3. And then we just ask if the CD8 T cells can kill the target cells that way. We measure this by looking at active caspase-3, which is downstream of pretty much every major killing function that CD8 T cells will use, whether it's Fas ligand, TNF, or perforin granzyme. Caspase-3 coalesces at the bottom of that apoptosis pathway. And although we can absolutely see that the CD8 T cells can mediate this killing, as shown in the example flow plots in the center, whether it's lymph nodes in the center of those flow plots or spleen, we can see caspase-3 induction by these CD8 T cells. Importantly, in the lymph node of the FTY treated animals, we do not see on the right that there is any difference in the FTY treated animals compared to controls. So again, there's no additional induction of cytotoxic activity beyond what was already present in the CD8 T cells, despite re-exposure to antigen. As I've kind of hinted at through this process, the study did not give us control of viremia. And as you can see on the top plot, when we interrupted ART, the FTY treated animals rebounded at the same rate as the controls, and in fact may have had, depending on how the statistics are done, have a slightly higher viral load compared to the control animals. If you look at the bottom small viral load plot, there's a trend towards increased viremia just at this one single time point that's shown. And if you compare the change in viremia from comparing the ATI peak to the peak of acute infection viremia, the FTY 720 treated animals actually have worse control compared to the controls. So what we thought was going to give us a benefit of increasing cytotoxicity in the tissues and had hoped that we would have better control actually turned into the opposite once we separate these, once we have the cells trapped in the tissues away from the cytotoxic T cells in the peripheral blood. To confirm that the CD8 T cells indeed in these animals were playing a role in controlling the virus, we went on and we did, after we had put the animals on ART following this initial treatment interruption, as you can see on the top, and we kept those animals on ART out to nearly a year post-infection. And then we performed a second interruption. And during that second interruption, we depleted CD8 T cells, as we can see on the bottom. And when we depleted CD8 T cells, we saw a very rapid increase in plasma viremia, suggesting or indicating that the CD8 T cells are indeed playing a role in controlling plasma viremia. But the FTY treatment during the first ATI indicates that the tissue specific CD8s are doing something different than what we thought. So with this in mind, we went on to ask whether we could modulate this. And so we turned to what the state of the field is using right now, which is PD-1 blockade and IL-15 superagonist. And these have been used individually in a number of different monkey studies, each of which have shown varying degrees of benefit towards viral control. So PD-1 blockade enhances T cell responses and viral control in a variety of models and also shown in SIV infection. And IL-15 receptor superagonist has been shown in a number of different studies to activate CD8 and NK cells to enhance perforin granzyme expression in lymphoid tissues and to also increase the number of cells that enter B cell follicles. And so we thought we were very clever, and we decided that we would combine both of these together, which has actually never been done in any study. And we thought we would hit these CD8 T cells in every way that we thought would be good. And so we did this, basically did a third arm of the study, set up in exactly the same way, with the ART initiated at the acute phase of infection, with FTY starting about 30 days before interruption. And during that initial phase of FTY, we started giving IL-15 receptor superagonist and anti-PD-1. This combined approach, there was some concern that maybe this would be sort of too much immune modulation, but this was safe and well tolerated, and we did not see any viral blips whenever we did this procedure before therapy was interrupted. However, we actually did not find evidence of control whenever we did this, and in fact, possibly a very slightly earlier viral rebound in the animals and a slightly higher viral load again, suggesting that trapping the cells in the tissues and modulating them with anti-PD-1 and IL-15 receptor superagonist actually has a negative effect on control, with slightly higher plasma viremia. Okay. We further looked to see if the CD8 T cells in the tissues responded to this therapy. And as you can see on the left here, when we look at SIV-specific CD8 T cells in the lymph node, either the GAG CM9 or TAT SL8 epitopes, we do see an increase in frequency of these cells, actually somewhat to high levels in the treated animals during the therapy interruption. But notably, we also, again, did not see, except for one animal, an increase in cytotoxic properties during this procedure, implying that despite what others have shown under the conditions of this study, we did not enhance cytotoxic properties with the combined immunotherapy in this model. So how do we think about this data then? So this tells us that cytotoxic function in tissues is not simply controlled by trafficking or lack of exposure to antigen, and suggests that tissue-specific regulation must dictate the functional properties of the CD8 T cells. We can say, because we've trapped all the target cells, all the infected cells in tissues, that we can say that plasma viremia in these SIV-infected animals must originate from the tissues, with the virus trafficking through lymph to get to the blood. We can also say that tissue-localized CD8 T cells are unable to prevent, delay, or reduce plasma viremia rebound after therapy interruption. It's not saying that they're not contributing to control, because we haven't done that exact experiment, but they are not able to improve control by themselves. And this then suggests that these cells are unable to eliminate virally infected cells, or the infection propagates too fast for the cells. And also that this combined immunotherapy, of course, failed to provide viral control. So what do we do with this? And so how do we think about this now in the context? Because now this is suggesting that the vascular CTL are really important in control, whereas right now the field feels that the tissue CD8 T cells are really the critical part of control, and it's both must be very important. But we wanted to ask ourselves, really, what is it that the CD8 T cells are seeing? What's their in vivo target? And it sounds kind of like a silly question, but really the reality is we don't know a lot about what the target of the CD8 T cells in HIV infection, what are the characteristics of these infected cells, really. And that's because we know a lot about the virology of the reservoir, but less about the cellular biology. So for virology, we've interrogated to many different ways the sequence of the virus, whether it's intact, clonal, how it's regulated, looking at it over time and so on. But the cellular biology is a little bit less clear. We know, of course, the virus infects CD4 positive cells of different subsets. CD4 positive T cell subsets have had virus quantified in many different ways, and there are many different papers that say different subsets of CD4s harbor more virus or less virus. But we know that they're very rare. There's not no consensus on whether there's a particular profile of infected cells, and we really don't understand the genomic profile of infected cells very well. And this is complicated further by what we know about CD4 T cell heterogeneity. And so, as I said on the last slide, depending on the study, many, if not most, CD4 T cell subsets have been shown to be preferentially infected. It just depends on the paper that you read. But for HIV cure, when we think about all these different subsets of CD4 T cells, every single one of them has unique characteristics that all independently are critical in the reservoir itself, because they affect the longevity of those T cells, their location and tissue trafficking ability, as well as how prone they are to being reactivated. And this applies both to blood and tissues. And so this is actually the reality of what the CD8 T cells have to combat. But we just didn't feel comfortable that beyond the obvious that this is the case, we didn't have a way to really understand those cells at any molecular level. So the question we had is, can we identify these cells from primary samples? And we wanted to do so without activating the CD4 T cells to induce viral gene expression, because once you activate T cells, you change those T cells in many different ways that can be unpredictable. And so we settled on using the presence of the integrated HIV-1 proviral DNA within every infected cell. We decided that would be potentially a molecular tag that would allow us to identify infected cells without activating them. And so the concept of this was to use single cell ATAC-seq, which I will explain on the next slide. And we settled on a strategy to align the data that we generated from this genomic procedure. We would basically just align it to both human and viral sequences that would allow us to directly identify the infected cells. And I'll sort of explain how that works next. And so ATAC-seq basically works by using a transposase that cleaves and tags open DNA. And so the idea here was that if that open DNA across the entire host genome includes the integrated HIV-1 provirus, that we could cleave that and tag it, and then allow us to identify it in our sequencing reactions. We then combine that with what's called select antigen profiling. And this is essentially just like very fancy multi-dimensional flow cytometry. It uses antibodies just like flow cytometry would do, but instead of using fluorophore conjugated antibodies, you use oligonucleotide conjugated antibodies, so that when you bring your sequencing through, you now have unique tags that corresponded to the presence of the antibody on each cell. And then you can essentially do flow cytometry by sequencing. That's the best way of thinking about it. And the advantage of this strategy is that because you can use unique oligonucleotide tags that can be quite long, you can multiplex these as long as your budget is for as many times as your budget is large. So in this case, we were using 154 different specificities of antibody to try to get an understanding of what is the cell surface profile of an infected cell. This combined procedure is called ASAP-seq, and that's what we'll call it going forward. We published this main work in Nature Immunology at the end of last year. So please feel free to look at that manuscript. I'm only going to talk about a small portion of the data today because it's too much time to go through it all. But basically, we can take direct ex vivo samples, or we can use in vitro infected cell lines. We use a 10x genomics based procedure to do single cell ATAC-seq or ASAP-seq, because we have both the ATAC and the antibody tags sequence. And then at the end, we're able to understand both the viral infection, the epigenetics of the cell through the ATAC, as well as the cell surface components. Just to sort of show what this looks like, I'm going to just focus on lymph nodes for here. This is a part of the data in the paper, but it's the piece of data that's relevant for the first part of the talk that I presented. So here, focusing on lymph nodes from 2 individuals who were off therapy. We basically just simply started with CD4 positive memory T cells, and we performed a procedure that I introduced to you prior to this slide. When we sequence the libraries that come out and we align them to reference viral sequence, in this case HXB2, you can see here on the right side a series of plots, and I'll describe what those are. The top part of the plot, every row that you can see is one individual cell that we found that contained an alignment corresponding to the HIV provirus in that individual. And in the orange areas, the orange dots or lines on there are the actual reads that we found, and the blue are the pieces in between the reads if the reads were close enough, allowing us to say that the whole section is probably there, even though we didn't sequence the whole section. And if you look at the small plot on the towards the bottom, with that looks like little mountains going across, that's showing the read recovery across the proviral genome, and you can see the map of the proviral genome on the bottom. So you can see the different sections of the virus that we're able to recover. This is just a way to make you comfortable that we can confidently identify an infected cell. We can then take those cells, and this is now taking the epigenetic data generated by the ATAC-seq and allowing us to identify the cell types. And so this is actually from in vitro infection. I'll show you the lymph node infection in just a moment, but this is to orient you. On the left, we have a UMAP projection of the different cell populations identified, and you can see where we've called what type of cell those are. So whether it's central memory or activated cells and so on. And on the right side, you can see for every individual cell that we profiled, whether or not we found an infected cell. And this is in vitro infection. This was an easy thing to do, just CD4s that we infected in vitro for a couple of days with virus, just to show that we could find the virus. So this is really just to orient you to how this looks. So the red dots are the infected cells, the gray dots are the uninfected cells in the same culture. And so really what we wanted to know was what do these virally infected cells in the lymph node look like? What type of cell are they? And so here now is data directly from the lymph node, where we can identify the different subsets of CD4 cells and other lymphocytes that we identified. So I could call your attention to the large green blob at the top, which is T follicular helper cells. These are cells that have previously been shown to harbor virus in infected individuals, but we also see other subsets of CD4 T cells as well. If we then project those cells in the red dots that have presence of virus, we can see exactly what type of cells we have. So we have T follicular helper cells, about 65% of the infected cells in these 2 individuals that are combined here. We have a variety of other subsets of cells that some of which, a smaller proportion, also have some virus in them. So this is the type of cells that the CD8 T cells will be interacting with in the tissues. We can look at different markers on these cells to get an idea of sort of what their antigenic structure is like on the cell surface. And we can look to see if there are particular markers that are higher than their uninfected cells. So if we look first here at the HIV negative cells versus the HIV positive cells, we see this marker called CD71 is highly enriched on HIV positive cells. CD71 is transferrin receptor, and we know this is upregulated on activated cells when those cells enter cell cycle. A number of other markers here as well. But really what I showed you on the last slide was that it's not that all memory CD4 cells are potentially infected. There's a distribution where T follicular helper cells are more likely to be infected. So the beauty of this procedure that we're using is we can go in and very specifically say, well, what's different about TFH cells? And if we just only focus on TFH cells, whether they're infected in red or uninfected in gray, we can say, are there any markers that are different about the infected TFH cells compared to the uninfected? And out of 154 markers that we labeled for on the cell surface of these cells, the only single marker that was significantly different on the infected TFH cells compared to the uninfected TFH cells is CD71. And that's most likely because these are, although most TFH cells are activated by nature, these must be the most activated TFH cells because they're the ones that are actively in cell cycle. And the other thing that we found here was going back to the residency theme from the first part of the talk. As I mentioned before, this the infected cells in tissues may be resident. When we looked at CD69, which is associated with residency, we can see that whether we look in HIV infected lymph nodes, or we've also adapted this assay to use in SIV. If we look in SIV infected lymph nodes, we can see that there's a higher level of CD69 on the infected cells, again implying residency and trapping of the cells in tissues naturally. We were also able to interrogate the regulatory structure of these cells by using the ATAC data. Remember, this selectively tags open genes, and so these open genes or promoter regions ahead of genes would tell us that these are genes that are sort of open and ready to be expressed or are being expressed. And when we do that, we can see in the lymph node that the uninfected cells are more likely to express they're more likely to have accessibility in their across the genome for the transcription factor GATA1. And GATA1 has been shown to directly repress CCR5 expression. And as you know from HIV entry biology, CCR5 is the co-receptor for HIV. So the HIV negative cells are essentially more protected because they are expressing they have higher GATA1 accessibility, and we know that GATA1 can repress CCR5. In contrast, if we look at the HIV positive cells, we see that these cells are much more prone to activation. And this is because they have a higher accessibility for the AP1 related transcription factors, including FOS and JUN, which we know are very prominently activated when the T cell receptor is triggered. And we also saw a large number of what I call POU-XF transcription factors because so many of them came up. We don't quite know the relevance of this. There are some papers that family members of this transcription factor family are involved in HIV replication, and we also know that there are binding sites for these transcription factors directly within the viral genome, but we still have to work through some of the relevance of this. So if we think about the properties of these infected cells that are going to be targeted by CD8s, we have a large, it's comprised of a diverse set of activated phenotypes that are going to be very prone for reactivation through sort of T cell receptor triggering. It's highly skewed towards resident characteristics, implying that these cells are not going to really be found in the blood very often. We found the same result in SIV infected lymph nodes, but we haven't done the key experiment, which is to look in ART treated lymph nodes. So we're in the process of doing that now. And so this brings me to my last conclusion slide, which I think is fun for discussion. And that is, how do we integrate all of this into a model where we could potentially develop a cure-based strategy. And so I envision this as sort of requiring 4 steps, and each step may have multiple steps. First, of course, we need a very potent and strong latency reversal agent. We don't have one yet. There have been many tries at doing this, but that's the first step. We have to be able to reawaken proviral expression. And that LRA, latency reversal agent, has to be able to reach cells in tissues. Otherwise, it will not be effective. So once we have that problem solved, then we have to modulate target cell access and elimination in the tissues, because most of the reservoir is going to be in tissues, not in the blood. So we have to either increase cytotoxic function of the resident memory T cells or increase the cytotoxic function of the migrating CD8 T cells that go into tissues. And as I showed you in the first part of the talk, that's not going to be easy, because just by exposing those cells to infected cells or by adding anti-PD-1, IL-15 receptor superagonist, we are unable to modulate that cytotoxic function very easily. So additional strategies will have to be devised there. We could engage or enable cytotoxic activity in the tissue sites very related to the CD8 cells, or alternatively, we need to figure out a way to mobilize the circulating cytotoxic cells, which normally can't get into tissues. We need to figure out how to get them to leave the blood and go into tissues where they could access the reservoir. The third part of this would be the opposite of the second part of this, and that would be not to try to move the CD8 T cells, but would be to move the reservoir. So can we modulate the reservoir to flush it out of tissues and into the blood, where it could then be accessed by the circulating cytotoxic cells? And we may have to enhance the circulating cytotoxic cells in the blood. We know different ways to do that, and maybe anti-PD-1 and IL-15 receptor superagonist would do that. I actually envision that both number 2 and number 3 would be necessary together. Well, not together, because you wouldn't do it together, but you would do one after the other. So you would modulate and switch, either pushing the CTL into the tissues or pulling the reservoir into the blood, and go through phases to allow eventually a decrease in the reservoir. And finally, and I think what nobody is thinking about in any of these strategies, but is crucial, is we have to assume that every time we try to modulate the reservoir, we're not going to be fully complete. And so we're going to potentially reinfect new cells during the process of trying to reawaken the reservoir and eliminate it. And so we also then have to include a component, and this is not art, because if you have art, you're not going to induce viral replication. You have to protect against new rounds of infection in some different way, so that you don't allow continual reproduction of your reservoir as you're trying to eliminate the reservoir. So with that, I will just go to my acknowledgments. This is the work of a number of different people in my lab. The first part of the study was conducted by Maria Pempena, a very talented postdoctoral fellow in my lab. The second part of the talk was conducted by Vincent Wu and Jamie Norden from my lab. And the monkey studies were done in collaboration with Mirko Paiardini at Yerkes National Primate Research Center, with collaboration with the others shown on the slide. And I will stop sharing and happy to take any questions. I hope that was all 100% clear.
Yeah, I think so. Well, not so much, because we have still so many questions. So we'll see. So
questions are good. Questions mean people are thinking about what I'm saying. So that's good.
Yeah, exactly. So yeah, I just would like to start with something from the first slides. So about the cytolytic function of CD8 T cells. So when we were reading your papers and so many others, for example, in the field of tissue resident cells, there is like some contradictory findings in some studies, you know. So we're trying to understand, I mean, how to reconcile this data. I mean, trying to see when you can find cytolytic function and when you're not going to find that. For example, we were trying to think about some variables that could explain that, like the experimental design, but also the age, perhaps, of the individuals, or perhaps a role of microbiota, or so many things that could be involved in this regulation of the cytolytic function in the tissues.
Right. I mean, the part of the problem is how do we define them cytolytic? How do we define them as cytolytic? I'm usually quite careful in saying that I'm talking about perforin and granzyme. And I think part of the problem, and perforin and granzyme together is essential. So part of the problem, I think, in the literature is that people often will just simply use granzyme B, and they will say that because there's granzyme B, the cells are cytolytic. We actually know that that is not true, and there's some very beautiful mouse studies actually which show that T cells secrete granzyme B simply to allow them to traffic through the extracellular matrix. That's part of how they travel through tissues, is by releasing granzyme B. Is not cytolytic activity. It's a trafficking function. And so when you just stain for granzyme B out of a tissue, you're going to see a lot of granzyme B positive cells, and people interpret that as being cytotoxic. But in the absence of perforin, you're not delivering that granzyme B to the target cell. You're just secreting the granzyme B, right? So there's a critical difference there. Another sort of partially controversial piece of this is because of one of our papers where we show that resident cells have a slightly higher level of perforin. They do so a little bit, but compared to a vascular cytotoxic cell, there's no comparison. They're much higher level of cytotoxicity of the peripheral vascular CD8 T cells compared to anything you ever find in a tissue resident memory cell. And then I guess the final piece is that in the mouse models, investigators have been able to drive cell function in TRMs. And depending on the state of disease of the animals they're testing, they can see these effects or not. We can't model that very effectively in humans. So I can't say that there's never, ever cytolytic TRMs. But we don't see them at steady state in HIV infection. That's the key thing, right? So people like to tell me that we're wrong because they can take cells ex vivo, activate them, and turn them into cytolytic cells. I can do that too, right? But when I take the cells out of the body and I look at them, they're not in that state. So that to me is the more important piece of data. The ex vivo or in vivo situation, and let's assume that those cells have interacted with targets recently, they're not differentiating to this state that people can do in vitro. So that's how that's how I argue with people about this.
You know, what do you think if you had the possibility to do the same studies in infants, you know, to see if perhaps, because, you know, we were thinking, we just have this hypothesis that perhaps the cytolytic function of CD8 and many, perhaps NK cells, I don't know, it's like an innate function, you know. So actually some studies of a group of Donna-Farber have found that actually cells from children have more granzyme B, for example, in comparison with adults. So I don't know, do you think there is something about the age that could be involved in this?
I mean, I don't know that Donna has done anything with HIV infected infants. I don't think so.
No.
I'm actually surprised by what you tell me. I haven't seen this reference myself. I mean, usually in infants, there's a very small subset of effector memory or effector CD8 T cells that usually takes time to accumulate. And so those of us who are old, like Manuel and I, have many more cytotoxic cells than you do, Federico, because you're young. So, you're the infant compared to us, and so usually one would expect. So I'm surprised, but I have to, if you can send me the reference that you're referring to. I'm
Okay.
not familiar with Donna's findings here. I mean, we've looked at a lot of actually, we've looked at some infants in our. We have other studies on type 1 diabetes, and so outside of the context of HIV, and we often get samples from infants. Their peripheral blood circulating CD8 T cells really don't have very many cytolytic subsets. Their lymph nodes do not have cytolytic subsets. We've looked in those lymph nodes as well. This would be mesenteric lymph nodes, pancreatic lymph nodes. We don't see cytolytic cells there either. T1D infants, you do see them. So that's a difference, but not in the normals. Yeah.
Okay. Professor Franco, you want to continue perhaps with the questions?
Yes, maybe I can pitch in. I just put in two of Donna Farber's references in the chat that might be useful. I would like to try and understand the circulation of these CD8 T cells. And in your Cell paper, the beautiful 220 paper, you show that CD27 in circulation could separate the two subsets, those that are not circulating, those that are circulating. And I would like to see if you could elaborate on that, and if possible, if you have studied homing receptors on those populations of cells, tissue homing receptors.
Yes, we have done the latter, and that was actually, I think, a supplemental figure in the paper, but we continue to interrogate those in a variety of situations. It's very clear that the trafficking subsets of CD8s in the lymph have heightened levels of tissue trafficking receptors compared to what you would find in the blood. That's very clear. I honestly don't know why CD27 marks the cells so nicely that have recirculation potential, but the association is very, very clear. The ligand for CD27 is CD70. It's a sort of a member of the TNF receptor family. There is signaling that occurs there. At one point, we did a long time ago, and this is all unpublished, but we have associations with the transcription factor TBET and CD27, which probably is important here. Like I said, we never published these data, but it's very clear that TBET. We think actually that TBET expression may downregulate CD27, would be, or at least is involved in downregulation of CD27. We've not shown that particular, but there's a very clear association between presence of the TBET transcription factor and the absence of CD27. It doesn't have to be, regulating one another, but until we show that. But those mark the differentiation states also very well. So TBET high cells are really the cytolytic cells, and they're always 27 negative. And we can see some of those associations then fall out when we look at the epigenetic profiling of the cells as well, in terms of those markers. The one curious thing that, again, not published, it's, as you know from having a lab, there's probably more data unpublished than published. We've from the 2020 paper where we did all the lymph studies, we actually have a very nice, mostly written story about the CD4 T cells in lymph that are in the HIV infected individuals. And one of the striking findings there was that the CD4 T cells, and this is not necessarily the infected CD4 T cells because we didn't test that, but when we look at CD4 T cells in the lymph of HIV infected individuals, whether they're on therapy or not, there's actually a marked decrease of CCR5 expression. We don't understand why that is, but it's a pretty striking finding, and it's the reason why we haven't published this because we really don't understand it. We have to get more samples to understand it.
Okay.
Any questions from the public, Federico? I don't see any.
No, I guess not yet, but we can continue here. So, I was watching your talk, and of course trying to contrast them with the papers. So I remember your cell reports paper in 2017 when we nicely showed that when you stimulate the cells with the peptides, the cells actually increase some granzyme B perforin, but also acquire some markers to egress from the tissue, right? But still, you don't see that in your study with the FTY, right? When you are trying to put in contact the cells with the antigen, but still they do not become cytotoxic. So why is that?
I mean, the in vitro studies from the cell reports paper were giving antigenic stimulation at a very non-physiological condition, and of course we cannot be 100% certain that the CD8 T cells that are trapped in the lymph node during FTY are actually all encountering a target. Is it possible we're missing some things? I could invoke a hypothesis that in fact there was a differentiation to effector cells in the lymph node, but those cells die because that's what effector cells often do after they're triggered. But if that were true, I would expect to see some, because we would be capturing them at a variable state, and so you wouldn't expect it to be so low that you wouldn't see it. So I don't think that's happening. What I believe is that there are counter-regulatory conditions in the lymph node that are not reflected in vitro that actually prevent the activation into the effector stage. So one of the key candidates for that would be TGF beta, which we know is very high in tissues. And there's also IL-10 could be a player here. And either of those have been shown to repress cytotoxic activity. And both of them are known to be in these tissues. So I think what's happening in vivo is counter-regulation of acquisition of effector function. And that's just not reflected in vitro because we didn't know about it then, and we haven't done those experiments. But I think there's data out there that suggests that this is the likely case. So that would be how I kind of put those two things together. Really the problem here, Federico, is that paper was published in 2017, data that was generated in 2014, right? So that's a long time ago, and now this paper is 7-8 years later, and so we've learned more along the way, and that's just sort of the way science goes. We evolve the way we have to think about things. What
for example,
a nice review that brings it all together. This is what we saw then, this is what we see now, and this is why you might think it's really different, but actually, it kind of still all fits. I'm not going to write that review. That can be for you. Reviews are a good thing for a junior person to write.
Dr. Betts, you mentioned that one of your ideas was how to get the activated cytotoxic circulating cells into the lymph node, getting them in there. But won't they just turn off? I mean, don't we have to find out how they're turning off?
So it is absolutely possible that they would turn off. Okay, so we did a whole nother study that's actually going to be in our paper on FTY that I didn't talk about because of the time. that is a study where, remember in the beginning of my talk, I mentioned that we know that cytotoxic cells are lost after the acute phase of infection in the lymph node. So we published that in PLOS Pathogens a number of years ago, and it's been replicated by others now as well. And so we actually, the very first FTY experiment we ever did was to try to stop that from happening. So we actually started FTY during the acute phase of infection. We actually started it as early as day 7 post-infection, and we hoped to trap the effector cells after they were induced for the first time. Hoped to trap them in the lymph node so we could see them, right? And we kept the animals in FTY out to day 90. So we thought, if they're there and we can make them accumulate, we would be able to tell if they're really lost or what's going on. And the answer was they were present early, just as we knew they would be, but cytotoxic function was gone by day 90, just like usual, even if we were trapping the cells in the tissues. The important part there is the SIV specific cells were still present in the tissues. So it wasn't that we lost all the responding cells. It's that the cells changed their expression. And so the acute burst of naive to memory transition, when the cells see antigen for the first time and they're activated, they definitely go through an effector cytotoxic phase, and that can be found in the lymph node. But after that priming and expansion event, they just progressively lose effector function. And some of it's probably egress, but it's not all egress, because that's what the FTY did, was stop that egress, and it was not sufficient to retain that function. So I think that there's this regulation taking place. Did that answer your question?
Yes.
That would have been about 10 more slides to take you through that.
I have another one. So you didn't talk about this, but in your paper about tissue resident cells in HIV individuals, you showed nicely that basically the lead controllers have a higher frequency of tissue resident cells, right, in the lymph nodes. But and also you said that probably is the antigen, the exposition to the antigen, that is inducing this tissue resident phenotype in these cells. But if the lead controllers, they basically do not see an antigen in the lymph node because they are controlling we know that there is some level of expression of antigen. I know. But how can be this occurring? And also the other part of the question is that you also showed that the individuals that had HLA-B57 have a tendency for a higher frequency of tissue-resident memory cells. So I was thinking if perhaps this is something to do with the TCR, and actually there are some papers indicating that these naive cells are predetermined to become tissue-resident memory cells. So what do you think about that?
Elite control. It's a long answer. A long answer is necessary because you have sort of multiple levels of things there. But first of all, not all elite controllers have good CD8 responses, as you know. Some of them have very few CD8 T cells, but those that do, I think there has to be ongoing antigen exposure to maintain these cells, because there just has to be. And I think if you look hard enough, you can find it. And that's been shown by a number of studies. For whatever reason, maybe there's just a lot of suppression happening and it keeps the virus level low, or maybe it's because the long-term non-progressors do have more cytolytic cells in their blood. Maybe there's a larger component of infected CD4s that traffic and get accessed by the cytotoxic cells in the blood of the elite controllers. So that's possible. So there's a bigger drain on the infected population compared to a progressor. That's a bit of an out there hypothesis that we have right now, and we don't have an easy way to prove it, but we're thinking about that. With regards to the TCRs, I don't know. it's so hard, Federico, because the whole antigen recognition requires TCR, and of course TCR is going to be important, but is there something special about TCRs for B57? Well, they interact with B57, so that makes them different than other TCRs. It's hard to know that there's really something very different besides the fact that in B57, the virus can't escape as easily because of structural constraints on the epitopes, just like B27. There's multiple layers to address that. But I'm honestly not. I'm still not convinced that differential TCRs are the answer to any of this. I don't find that as important yet, because there's much larger differences in just the regulation of CD8 T cells across these sites and their function, rather than getting down to like this molecular, it's all about one single TCR, because I just don't, that's not true, because the functions are across TCRs, the functional differences in the subset regulation is TCR independent. Yeah. I think we got to solve that part first, and then we can get to the TCRs.
Profe, you have another question? Profe Franco?
No, I was thinking that we would get some questions by the YouTube, but I think we are over those. We don't have any more there.
Okay.
Perhaps I just would like to go to your last slide before your acknowledgement slide regarding the therapeutic approaches that we could use to try to
you want me to put it?
HIV.
You want me to share it again?
Yes.
Well, yes, it could be. So it's basically, I mean, you mentioned the potential steps that we will have to do, right? But how, which specific
Can you see this?
you suggest? No, just.
Well, I mean, each one of them has, can you see the slide?
Yes.
Yeah, so I mean, each one of these is complex, right? So, yeah, Manuel, I remember now you had mentioned asking about this. So, for example, if you take the question about mobilizing circulating cytotoxic cells in the tissues, that's what you asked about.
Right.
The issue is that CTL, this subset, does not express CD62L, which doesn't allow them to, interact with the high endothelial venules that would let them get into lymph nodes. They also don't express CCR7, which prevents them from being attracted to the lymph nodes. So, one or both of those molecules would have to be re-expressed on the CTL. Whether you add it exogenously somehow with some type of vector or genetic strategy, or you try to reverse the regulation that's suppressing or the expression. In these cells, the locus becomes in heterochromatin for these genes. So I mean, it's an epigenetic regulation occurring that turns these off. So we would have to bypass that somehow to do that. The same is probably the case for increasing the cytotoxic functions of either of those populations, because the problem is, for example, the tissue migrating cells are in an earlier differentiation state. So if you differentiate them to become cytotoxic, they're going to lose their tissue migrating capacity. So like I said, none of this is easy, but this is the only way to change it. And so we have to figure out a way to bypass the natural biology of these populations in order to manipulate them.
Exactly. Now, about the 9 superlytic functions, that is like a very hot topic now in HIV. What do you think about it? Do you think it's better to try to promote the cytolytic function and repress the non-cytolytic and inducing the latency of the cells, or trying to promote both? What do you think about it?
That's a difficult one.
So I think it depends on what your strategy is. If you want to eliminate the reservoir, the suppressor function is not what you want. If you want to go for the block and lock strategy, where you permanently repress viral reactivation, suppression is the way to go. I think that in the field we would prefer to eradicate the reservoir rather than let it stay in place and hope we can maintain suppression. That's I think a more risky strategy to go after, unless you don't want to use a therapeutic, because then you might as well just stay on art, right?
Yes.
Anything that sort of maintains a virus in a permanent blocked state is just another form of art. So people don't want to take drugs every day or have to get shots every few months or whatever the long-term strategies are. People aren't going to want to get bean abs, infused to try to repress that. That's worse than taking art. They want to eliminate the reservoir, and I think you have to go after straight up elimination. Merck has a new compound that seems to selectively target cells with active virus and eliminate them, but we don't know whether that product will eliminate latently infected cells. It probably won't, because it seems to target cells because they have viral proteins. So, yeah.
Okay, I think we have plenty of questions. I don't know if I think we don't have more questions by the audience. We can start closing now. So,
Okay.
Dr. Betts, again, thank you very much. It was a great pleasure. I think we all learned a lot today. So I hope to see you again. We still stay in contact. So as you know, I'm very interested in the field. So of course, you will know about me many times.
Yes.
Thank you
It's
very
been
much.
a pleasure. Thank you very much for the invitation.
Thank you, Dr. Betts.
Diana.